Hello, so in the last video we discussed mitosis and the cell cycle in general. But now a question still persists. Well, in mitosis we get two genetically identical cells. Well then, how are gametes created, which merges into the zygote, which has ability to divide mitotically? Well, it happens through the process called meiosis. Here we get the gametes. To explain meiosis in deep, we first have to check what is the ploidy of the cell. So ploidy tells us how many complete sets of chromosomes does organism have. For example, here it is the diploid set because each distinct chromosome repeats twice. But here each one of them repeats only once, meaning that it is the haploid set, so it is the haploid cell. Now, as we can see, if we merge two haploid cells, we get a diploid cell, which can actually divide mitotically. As such, it is logical to assume that meiosis is a way which gives us a haploid cells from a diploid one. And let's begin with meiosis. Here we have the normal cell with DNA already condensed, which happens into the prophases, of course. But the meiosis consists of the two parts. It is the prophase 1, so meaning that these numerations are the meiosis 1 and meiosis 2. So meiosis 1 begins with the prophase 1. Here a very interesting thing happens. So let's take two homologous chromosomes with two sister chromatids, and they actually not merge, but they actually hold each other and create a structure called bivalent. Now, after creation of the bivalent, very interesting thing happens. They have ability to exchange genetic material between each other. They will exchange it, and this process is called crossing over. As you can see, with homologous chromosomes having ability to exchange genetic material with each other, it increases the genetic variability which the offspring will have. So for example, there will be a different combination of alleles which could be generated into this way. And after meiosis 1, they of course get separated, but we will get to it closely. Well, here the bivalents got created, crossing over happened, the nucleus gets destroyed, the centrosomes move to the opposite poles, the microtubule spindle starts to assemble, and this is when the prophase 1 finishes. In metaphase 1, as we mentioned before, the actual bivalent structures are now aligned to the middle of the cell, to the metaphase plate. And after each structure is connected, after each chromosome is connected to the centrosomes, the anaphase starts, the first anaphase, where the homologous chromosomes are brought to the opposite sides of the poles, and in telophase 1 we get two cells, and here the meiosis 1 finishes. As you can see at the start of the video, in the prophase 1, the starting cell had two repeats of each distinct chromosome. Here each cell has only one, meaning that we already got two haploid cells. Only thing left to do is to separate sister chromatids from each other, which is really similar to the mitosis. Here we have the prophase 2, we have the very small interface here, and again all of the structures get disassembled and the mitotic spindles get created and everything repeats again. And in metaphase 2, of course, the chromosomes align to the metaphase plate and they are connected to the microtubules. After that, anaphase 2 starts. Here the individual chromosomes are separated from each other to the opposite poles, and in the end, in telophase, we get 4 distinct gametes. And here meiosis two finishes. As you can see, we got four different gametes with having a different genetic variability due to crossing over or combinations of different alignments over the metaphase plate. And after further modifications, we can either get the egg cell in females and the sperm cells in males. Well, we came to the end, and in conclusion, meiosis is the mechanism that ensures to create gametes, haploid cells, from a diploid cell. And meiosis ensures the genetic variability through the mechanism of crossing over, allowing different combinations of alleles to be present into the various offspring. Thanks for watching and see you soon.